They themselves are also called upon to present in person all around the world. So at this point, I'd also like to take this opportunity to thank our advisory committee who have taken time out of the day to present here today, as well as all the time behind the scenes to help with what we do with our Rett community. Just to give you an idea, last weekend on Saturday, this committee travelled from their home states all around Australia to convene in Sydney and spent almost 8 hours in a boardroom building the framework on the program for the 9th World Congress that the Rett Syndrome Association of Australia will be hosting next year. So with that, we'll move along to the agenda and some housekeeping rules, please. So we'll go through a welcome from Wendy. We'll meet the committee. We'll give a brief update on the Nashville conference, which was June earlier this year. We'll have questions and answers on any of the presentations. And we'll do a quick brief on the World Congress update. And then we will close. So just a couple of housekeeping items, if I may. So if everyone could please put their computers on mute. There was some talking coming through before, which is a little bit distracting for the presenters. I know a lot of the time it's unintentional, but yeah, if that mute's on right now, it won't distract the presenters from working. This meeting is being recorded. That was definitely mentioned in the mail out, and this will help any people that weren't able to attend today to view the recording. As for any questions, if you could please submit those through the chat, and then if we have time in the questions and answers, we'll submit some of those questions to the presenters. With that, I'd like to hand over to Dr. Wendy Gold to present. Thank you, Wendy.
Thanks, Claude, and welcome everyone. I'm very excited to be able to speak today on behalf of the Scientific and Medical Advisory Committee and to give you an update. As some of you may know or may not know, that we formed this committee in association with the RSAA this year, and we've already had 2 meetings. The committee is comprised of clinicians and researchers that are involved in Rett syndrome research, whether it's MECP2 or CDKL5 or FOXP1, due to their expertise in the area. And we've come together to form this committee with a number of aims and purposes, which I'm going to go through. So the committee consists of, which Claude had up on the screen, myself, Associate Professor Jenny Downs, Associate Professor Helen Leonard, Associate Professor Carolyn Ellaway, Professor John Christodoulou, Dr. Marianne Lancaster, and Professor Eric Gowans. And as I said, we meet 4 times a year. I've already had 2 meetings. And the overarching aims is to support the Rett Syndrome Association of Australia and the Rett Syndrome community in Australia. Now, specifically what our aims and purposes are is to provide non-binding but informed guidance on matters specific to Rett Syndrome, scientific, health, and medical strategies, and this includes clinical trials. Another aim is for us to work with the RSAA committee to identify partnership opportunities. We also aim to support research grant applications that are submitted by the RSAA to other funding bodies to help support the RSAA and the communities, to contribute to any relevant RSAA media and advocacy opportunities, and also, lastly, to assess the merits of any future funding applications provided by the RSAA and make recommendations to them to whom the grant should be awarded. So this is a very broad overview and reach of the committee, but we hope the aim is to support the Rett Syndrome Association of Australia and also the community in Australia too. So with that, I'm going to pass back to Claude.
Sorry, Wendy, we might proceed to the next slide and have you, I've just actually managed to access your slides. So if everyone will bear with me.
Thanks, Mary. So what we're going to be doing next is we're going to give an overview of our research and our clinical work. related to our position on the Scientific and Medical Advisory Committee, and I've been chosen to go first, and I gave my slides to Mary very late. So this is my fault for these coming in now. So thanks, Mary, for being so accommodating.
No, it's not a problem. Can we see it now?
Yes, I can see
Great.
it.
Okay.
Okay, so that was on the Scientific and Medical Advisory Committee, which I've spoken about. So I'll just briefly chat about my research, which is on the next slide. All right, there we go. Thanks, Mary. So I thought I'd break this down into who, what, where, and how. So the GOLD lab, or the Metabolic Research Lab, is and now the Molecular Neurobiology Research Lab is based at on the Westmead Hub, which is in Sydney, in Western Sydney, and we're actually physically located in Kids Research, which is the research facility of the Children's Hospital at Westmead. And also, I have an affiliation with the Children's Medical Research Institute. People know them as the Genes for Genes Institute, where we work very closely with researchers there in collaboration for developing the studies that we are doing. Thanks, Mary.
Sorry, there's a little delay with moving the slides forward for some reason. Oh, there we go.
That's okay. All right. So who are we? This is a picture of some of my team. We have in our team, we've got a multidisciplinary team of 2 postdoctoral scientists, a handful of two handfuls of PhD students, research assistants, and undergraduate students. So as you can see, it's a mixture of very intelligent and energetic people that help drive our research forward. This is a picture taken last year of just a couple of us who were able to make the photo opportunity. And of course, with research, there's a lot of flux in the people that are in the research team at any one time. And then my next slide is what we do. So there are a number of things that we do. First of all, I've broken this down into pillars. One of our big research projects at the moment are biomarker studies. And the reason for this is that the causative gene for Rett syndrome, MECP2, was identified, or mutations in the gene were identified in 1999. That was a long time ago. And since then, over 60 clinical trials have been conducted, and there hasn't been a therapeutic specific to Rett that has entered the clinic until March this year, which is trofinetide, and it hasn't come to Australia yet. So we've been wondering why this is so. And one of the reasons, not the only reason, one of the reasons is there are not easily identified clinical biomarkers that can be identified that specifically define Rett syndrome or diagnose Rett syndrome patients. So we thought that we would have a look to see if we can find any biomarkers. These biomarkers could be for prognosis, disease severity. They could be targets for novel therapies, but also specifically they could actually land up being endpoint markers for clinical trials. So we're really excited about this project. We've got some funding to do this at the moment, and we're excited for the outcomes. The other thing we do is we use modeling. We have lots of modeling studies. We do have a mouse model, but we've now moved more into using induced pluripotent stem cells, which are stem cells that we reprogram from patient blood, and we grow these into mini brains or neurons. The mini brains are nothing like a brain at all. They look more like a McCain's pea than a brain, but they have neurons, which are brain cells that actually connect and work together, and we use these to test on novel therapies. We also have a drug discovery program as well. And then lastly, we also have a gene therapy program as well. And we are developing different gene therapies for girls that have got mutations in MECP2. And segueing into from this is I just want to highlight a bit about the gene therapies that are very topical at the moment for Rett syndrome. Which is on the next slide. The first one is the Taysha gene therapy. They've dosed the first patient in Canada. Claude might be mentioning this from the Nashville talks. And then there's another gene therapy, which is by Neurogene, and they've announced the locations and enrollment for gene therapy in the US. So this is all very exciting. And as I said, we're also developing some gene therapy. So we know we know very well, we're very excited about this space. Now, my last slide, what I wanted to show is why gene therapies may work. I've shown that in the video, they're two mice. One is a mouse that does not have MECP2 expression, so it is indicative of a patient that would have a mutation in MECP2. And as you can see, it's sitting very still. The quality of the images, the videos are not very good. It's sitting very still and it's not moving a lot, and it has some phenotypic resemblance of a patient with Rett syndrome, but obviously not completely, because it's a mouse and not a human. Now, the mouse on the right-hand side is the same mouse, but it has had MECP2 reintroduced into the body. It's not a gene therapy at all. These studies were done by Adrian Bird and Jackie Guy in 2009. It's not a gene therapy. It's a genetic manipulation, which can be done in humans. But what I wanted to show you is that these studies show that it is possible that there could be a reversal by gene therapies. What I also want to say is that gene therapies are still very investigational, and they do come with high risk as well. So it's not a therapy that is needs to be taken lightly, and there's going to be a lot of consideration into the into ensuring that the gene therapies that are coming through for Rett syndrome are not only effective, but they're safe as well. So that's all I had to say. Thanks, Mary.
Thanks, Wendy. I'll just bring up our slides for the rest of the presentation. Lord, we might now hand over to Dr. Mary-Ann Lancaster and Professor Eric Gowans.
All right, I'd like to introduce Mary-Ann Lancaster, who's a Rett mum and part of our committee, as well as the scientific committee, and Professor Eric Gowans, who is also part of our committee and the scientific committee, with who has a Rett granddaughter. I'll pass over to you both just to give us a bit of an introduction. Thank you.
You can go first, Eric.
Thank you, Mary-Ann, and thank you everyone for joining us. As Claude has said, I'm the granddad of a beautiful 8-year-old girl who has Rett syndrome, who was diagnosed 5 years ago. Soon after that, my wife and I found that there was a scientific meeting in Rome on the syndrome, and we went to that meeting in Rome, where we met Tony Cagliuso, who's the treasurer of RSAA and was representing RSAA at that meeting. My wife and I joined RSAA then, and I've been a committee member since then. I'm now retired, but I was a virologist and molecular biologist for around 35 years, working on different viruses and vaccine design before I retired at the end of 2019. However, I no sooner retired than the COVID pandemic hit. I found that I was spending a considerable time on the phone to the people in my old lab, and it was easier to go back to work, so that I spent the next 3 years working on a COVID vaccine that was developed in the lab in the University of Adelaide, manufactured locally in Adelaide, and administered to volunteers in a phase 1 trial in the RAH. I retired again at the end of 2022. I now use my knowledge and expertise in molecular biology to keep up to date with new developments in Rett research, particularly gene therapy, as I previously worked with adeno-associated virus, and many of you will know that the AAV is currently the method of choice to deliver a functional MECP2 gene to patients with who are enrolled in gene therapy trials. I've met the scientists who lead this work, and I can discuss the strategy results with them. So in addition to my role as a committee member of RSAA, I also act as the chair of the scientific subcommittee for the 9th World Rett Congress that will be organised by RSAA in October next year. And Claude will tell us a little of that a bit later in the webinar. And finally, I also try to translate complex scientific results into lay language, then write short reports which appear in the RSAA newsletter and website to inform the RSAA community in Australia of recent advances that may eventually lead to new therapies for Rett patients. So thank you for your attention.
Thank you, Eric. I'm Dr. Marianne Lancaster, and I feel very intimidated being on the SMAC committee because I'm the only one who doesn't have professor in front of their name or associate professor. I'm a general practitioner, and I'm also the mum of a 25-year-old Rett girl. My introduction to Rett was quite different to a lot of people's, in that my daughter wasn't diagnosed until the age of 21, despite being at the Royal Children's Hospital from about the age of 18 months, when she began to regress her IQ, her hand movements. But because she was still verbal, and she still is, and she's still mobile, and she still is, she didn't seem to trigger anyone to think that it could be Rett syndrome. So it wasn't until her genetics were done at the age of 21 that she was diagnosed. So as soon as I found out about that, I got in touch with RSAA, who were fantastically supportive, and I decided that I wanted to be on the committee, which I joined straight away. So that is me in a nutshell.
Thank you very much, Marianne and Eric. And now we'll move, excuse me, on to Professor Helen Leonard. Helen, you'll have to unmute yourself, sorry.
Thank you very much, Mary and everyone. I think you could move on to my first slide, please, Mary. So I'm delighted to be able to present this recent research on Rett syndrome to the online audience here. And before moving any further, I'd like to acknowledge all my co-investigators on this study. Next slide, please. But before talking specifically about this study, I'd like to give a very brief background on the course of Rett syndrome up until now, for those who perhaps are not aware of it. So in 1966, Rett syndrome was first described. Next, please. But information about Rett syndrome didn't reach English-speaking medical community until 1983, when it was published by Professor Bengt Hagberg. Next slide, thanks. And then the first clinical criteria for Rett syndrome were published in 1985. Next. They were revised in 1988 by a US committee. And in 1993, we established the Australian Rett syndrome database. Not long after that, Professor Hagberg wrote a paper describing the atypical forms of Rett syndrome, which is still very relevant today. Next slide, thank you. And then in 1999, as you probably all know, the genetic cause of Rett syndrome was identified, a mutation in the MECP2 gene. The clinical criteria were updated again subsequent to this. Next slide. And then in 2003, we established the international Rett syndrome database called InterRett. Next slide. And then, as you've already heard, in 2007, Adrian Bird reversed Rett syndrome in a mouse model. Next, thank you. The criteria were revised again in 2010, and these are the current clinical criteria. And so I'd like to say that families in Australia and around the world have been participating in research over all this time. And as a result, children with Rett syndrome are being diagnosed generally earlier, and much more is known about the clinical variation, and guidelines for management have been published and are in use. So next slide, please. So just to mention the Australian Rett syndrome database, just briefly, this slide shows many of the people who were involved in its establishment and ongoing maintenance. Bill Callahan, you will see there, John Christodoulou and Carolyn, as well as Alison Kerr from Scotland, who was involved in the very early years of Rett syndrome. On the panel on the right shows you all the types of studies that have been carried out using data from the Australian Rett syndrome database, which has all been, a result of family participation. So thank you all. Now I'll move on to the topic of the study, which is the mental health. But we needed to be able to do this study, we needed to have an international database, because although we have population-based data in Australia, our number, it's a small country, and our numbers are limited. So to look at the true variability, we had to have a bigger sample. Next slide. So the aims of the international database are to provide a large sample of cases for analysis, encourage collaboration among researchers across the world, increase the clinical understanding of Rett syndrome, and provide an innovative and efficient mechanism to disseminate research findings and provide clinical support. So the background to the mental health study is that although Rett syndrome is associated with many medical complications, it has mainly been the physical aspects of the disorder that have been studied. And there has been very little focus on behavioral and mental health aspects of Rett syndrome. Next slide, thank you. So sleep disturbances and other behaviors such as screaming, social withdrawal, mood swings, self-injurious behavior may affect mental health in Rett syndrome. But other than a small number of recent studies, there's been very little literature on this. So our current study really aimed to examine the determinants of mental health and Rett syndrome using data from the International Rett Syndrome Database. Next slide, thank you. So what we used the Interrett database as our sampling frame, as I already mentioned. Next, thank you. We administered follow-up questionnaires in 2015 and 2018, and we used the data from the 2018 study in this analysis. We included questions on mental health. We used a scale called the Anxiety, Depression, and Mood Scale, which measures anxiety and depression and other aspects of mental health. Next, thank you. So this is the study participants. We had a bias towards older people because many of these individuals were recruited to the study in the early days. So by 2018, many were into teenagers or young adults. We had a good distribution of mutation types. And for functional ability, about a third were able to walk independently, a third to walk with assistance, and a third unable to walk at all. Next slide, thank you. We looked at other factors in terms of sleep. We used something called the DIMS, which is disorders of initiating and maintenance of sleep, and the DOES, which is disorders of excess somnolence. So one is for sleep disturbance, the other is more for excess sleepiness. We looked at the frequency of seizures, and we also looked at the presence of autonomic disturbances, both breath holding and hyperventilation. And then we had our scores on the ADAMS. Next slide, thank you. So we analyzed it statistically using linear regression, and we looked at the effect of age group, genotype, ability to walk, and sleep on the anxiety scores. What we found out was that the most of these domains had relatively similar average scores. However, we found that for higher anxiety scores were associated with those who had a milder phenotype or who had sleep disturbances. So they weren't the girls and women with severe Rett syndrome. They tended to be those who had a milder variation. We also found that in older age, the anxiety scores lessened. Next slide. So we found particularly that relative to one of the R270X mutation, those with a mutation called R294X scored nearly 4 points higher for general anxiety than the others. We also found that there was differences in the depression scores, but I won't focus on the depression for this short talk. Next slide, thank you. So just here you can see I'm highlighting the fact that the older girls had a lower, just go on, Mary, thank you, next one, next, thank you. These are lower scores for the older individuals. Next, please. And you can see that for the R294X mutation, the anxiety scores are much higher. Next slide, please. And this looks at the relationship between anxiety and other medical complications. And you can see here that for sleep insomnia, difficulty with sleep, this was associated with anxiety, as was excess somnolent daytime sleepiness as well. And the other association was with the autonomic breathing abnormalities. Both the hyperventilation and the breath holding were associated with anxiety. Next, thank you. And this just shows that the relationship with the R294X mutation graphically, you can see that the scores there are much higher than for the other mutations. And finally, my last slide, please. So our results are somewhat consistent with those from a very early study we did in 2006, but now we have much larger numbers, and we were quite confident in our results. We found that anxiety was increased in those with this R294X variant, but we previously also found a relationship between this mutation and quality of life and sleep and autonomic function, and they were all compromised in those who carry this variant. So really, this is the beginning of a body of knowledge which is gradually building on the mental health of girls and women with Rett syndrome. And we believe it's got implications for counselling, clinical management, and possibly further understanding of the MECP2 functioning. Thanks very much. And I'd like to acknowledge all the families who participated in our research and the International Rett Syndrome Foundation for funding this study. Thank you.
Thank you, Helen. Thanks for that insight on the natural history of Rett syndrome in Australia and your mental health study. I'd like to present Associate Professor Carolyn Ellaway to present. Thank you. Sorry.
Thank you very much, Claude. Good afternoon to everyone. It's lovely that everyone's together and getting an opportunity to meet everyone on the Scientific Medical Advisory Committee for the Rett Syndrome Association of Australia. So I'm a clinician. I'm a clinical geneticist and a metabolic physician. I have appointments at the Sydney Children's Hospital at Randwick and the Children's Hospital at Westmead. I also have a couple of private genetic clinics and more recently have started in the area of reproductive genetics and work with IVF Australia. But my journey with Rett syndrome started way back in 1996. I had just finished my paediatric exams and I was very keen to start training in clinical genetics. But at the time, there was no position available for training at the Kids Hospital at Westmead. And I'd already had a few discussions with John and John Christodoulou, and said, look, you know, there's no position for training. Is there a research project that I could get involved in, and maybe I could apply for a clinical fellowship with the hospital? And John just happened to say, well, I've been talking to Helen Leonard, and we're thinking about this clinical trial of a medication called carnitine in Rett syndrome. Would you be interested? And I went, yep, sure. At that stage, I had never seen a girl with Rett syndrome. I'd heard about Rett syndrome. And so I put in an application and I got a hospital fellowship. So I started a clinical trial of carnitine in a cohort of 35 girls with Rett syndrome from across New South Wales. Now, at that time, there was no clinical research group. So everything in relation to that clinical trial, I pretty much had to do myself, including liaising with pharmacy, recruiting families, organising the rooms where I could do the consultations, collected all the data myself, analysed all the data with the help of a statistician. And at the time, John Christodoulou was my supervisor. It was only meant to be a one-year fellowship and a short clinical trial, but that led to further and further interest in Rett syndrome, additional studies, including doing an ECG on all of the girls in the cohort. And we identified that around 25% of girls had a prolonged QT interval on the ECG. There was interest from the families to continue the carnitine after the completion of the double-blind placebo crossover trial. And so I then embarked on an open label study. And of course, this was another significantly large project. And at the end of all that, I managed to be awarded a PhD. And John was my supervisor for the PhD. We certainly got a lot of publications out of the work at the time, and the families across New South Wales were very generous in their time contributing to this research. So at the end of that, I said to John, look, you know, these families have been so generous, we should be giving something back to them. And how about we think about a clinic dedicated to girls with Rett syndrome? So with the support of the Rett Syndrome Association of New South Wales, we were able to get some funding, and we started a clinic dedicated to girls with Rett syndrome at the Children's Hospital at Westmead. That started in 2000. The clinic is still going. We have myself as the clinical geneticist. I'm also a paediatrician. I have a genetic counsellor, a dietitian, a dentist, physiotherapist, occupational therapist, speech therapist, and a music therapist at the clinic. And we've just completed a 20-year review of the patients that have attended that clinic over a 20-year period, and that's currently under review for publication. Since then, I've also been involved in a couple of other clinical trials. One was a clinical trial of medicinal cannabis in girls with intractable epilepsy and Rett syndrome. And then also I'm the principal investigator on the Anavex study, which that has completed in New South Wales. It's still ongoing in a couple of other states, but nearing completion. Over the last 25 years or so, I've had close collaborations with Helen Leonard, Jenny Downs, John Christodoulou, and Wendy Gold. We a dedicated group of clinicians and researchers to Rett syndrome, and really looking forward to our ongoing work with everyone on the group and also the Rett Syndrome Congress next year. Thanks very much.
Claude, you're on mute, sorry. I think Claude's got some technical issues. I'll just step in and introduce Professor John Christodoulou, who's going to talk to us now about his work.
Thanks very much for that. My background is I initially trained in pediatrics and then in genetics, where my primary clinical focus was involved in the diagnosis and management of kids with inborn errors of metabolism. I first became interested in Rett syndrome probably in the early 1990s, and Helen can correct me if I'm wrong, but it was through a chance encounter with Helen at a pediatric conference. And so we struck up a conversation and thought it would be a good opportunity to become involved in some collaborative research. So in the early days, which would be the mid to late 1990s, we were involved in trying to hunt down the gene that caused Rett syndrome. We all knew it was on the X chromosome, but and a number of groups were looking, and in the end, Huda Zogby and her group in the US identified the MECP2 gene as the cause. But because of our involvement, we were poised to rapidly start screening the gene in a pretty large cohort of Australian kids with Rett syndrome. So we performed research-based gene analysis in around 270 girls with Rett syndrome, and that formed the framework for then being able to start a long collaboration with Helen and Jenny and others in terms of delineating the correlations between the clinical features in Rett syndrome and the various genetic changes in Rett syndrome. Carolyn's mentioned the Rett Clinic. Along the way, with in collaboration with Patrick Tam at the Children's Medical Research Institute, we developed several mouse models of Rett syndrome that we studied. And Carolyn has already very generously indicated that she was a PhD student with me. Along the way, we've had an opportunity to supervise about another dozen master's or PhD students who were specifically focused on Rett syndrome. And along the way, we were actually involved in the discovery of the second gene that was thought to be causative of Rett syndrome, and that's CDKL5. At the moment, so I moved from the Children's Hospital at Westmead at the end of or beginning of 2016 from a clinical and research role to a purely research role at the Murdoch Children's Research Institute in Melbourne, which I'm now been based for the last 7 years. And among other things, one of the areas we're researching in is CDKL5, and the people who were involved in the last slide were Nicole Van Bergen, a really talented postdoc, Sean Massey, a research assistant, and Anita Quigley, who's based at St Vincent's Hospital at the Aikenhead Centre, and she's a fabulous mini brain or brain organoid researcher that we've been collaborating with. So people may not be that familiar with CDKL5, so I'll just to give a little bit of background about it. It's a genetic disorder where one of the key features is very early onset and severe seizures, developmental delay and intellectual disability. And we initially, it was thought to be a second Rett gene causing a more severe variant of Rett syndrome, but with studying a larger cohort or group of girls with mutations in CDKL5, we found a number of differences. Firstly, the girls had facial similarities, whereas girls who have mutations in MECP2 don't have any facial differences from the general population. And so this was part of work that was part of Stephanie Fair's PhD work, who was supervised by Helen and me. The next slide. In terms of what CDKL5 is about, I'm again just to highlight that the very early onset seizures are a predominant feature in that, and you can see there that whilst seizures occur in individuals with MECP2 mutations, they occur much earlier and are often much more severe in CDKL5. Next, thanks. And some of the other common features that we see in individuals with CDKL5 include reduced muscle tone, interrupted sleep patterns, feeding, digestive disorders, breathing problems, problems with vision, and poor muscle strength. So you can see that a lot of those features are very similar to what's been described in individuals with Rett syndrome, and that's why it was thought to be a variant of Rett syndrome. Next. And it so happens that the gene is also on the X chromosome. And so like in MECP2 related Rett syndrome, predominantly affects girls, although boys can be affected, and they're usually more severely affected. Next. So the CDKL5 based research that we're doing, a number of questions we're asking. What is the function of CDKL5 in human brain cells and in different compartments of the cells? Can we identify targets of CDKL5 which might then be give us pathways to targeted therapies? And can we develop more accurate models of the CDKL5 deficiency disorder for screening some of these new therapies? So next slide. So we're using approaches that Wendy has already outlined that her group has been involved in, and that is we can take cells from an individual with CDKL5 deficiency disorder, manipulate or trick them into becoming stem cells, which we can then push down to become or develop into brain cells, and then brain organoids or mini brains, as Wendy referred them to. And then we're applying a number of lab-based technologies, gene expression studies, protein studies. We can actually measure the electrical activity of the neuronal cells and also the mini organoids and demonstrate they have an epilepsy phenotype. And then in terms of doing all of that, the end game for us is to then use various approaches to see if we can either identify existing drugs that can be repurposed to treat individuals with CDKL5 deficiency, or through our basic biology studies, potentially identify new drug targets that could be developed. So that's the CDKL5 work we're doing in a nutshell. And just finally to acknowledge on the next slide, a number of funding bodies that have been helping to support our CDKL5 research. And that's all for me, thanks. And I'll hand back to Claude, and hopefully his technical issues have been resolved.
I think I am. Can everyone hear me?
Yep.
Seeing a lot of nods. Thank you. Thank you very much, Professor John Christodoulou, for the insight on CDKL5. I'll hand over now to Associate Professor Jenny Downs to give us some insight on her work. Thank you, Jenny.
Thank you, Claude. Can you hear me?
Yes.
Great, thank you. It's great to be here. It's great to have this webinar to connect with you across Australia, wherever you are. I can see names I know in the list of who's here, and that's wonderful, and names I don't know. And so it's great to meet you all. I've titled this Introduction to Me as a Knowledge to Practice Roadmap for Improving Quality of Life in Children and Adults with Rett Syndrome. I'm Jenny Downs. I work at Telethon Kids Institute. I've worked with Helen since 2005, and I'm now the Child Disability Lead for our team. Thank you, Mary. I started as a pediatric physiotherapist, just as my background, and I worked in Perth, in Sydney, actually at Westmead, and also in London at a few hospitals in London. And my last job in London was that as the head of a physiotherapy department in a small children's hospital in the East End of London. So that was a very colourful, exciting place, but then back to Australia. And then following writing a PhD, I was lucky to have opportunity to start in a research position at Telethon Kids Institute. I started with Rett syndrome. This was 2005, working first on motor skills, that we were evaluating videos and developing that into how we could score those videos. And many of you will have contributed videos to our study. And beyond that, there's more work on fractures, scoliosis, gastrointestinal issues and gastrostomy, early development, and way more. And so that is a summary of Rett syndrome, but there's also been extensive research into other rare diseases, including the CDKL5 deficiency disorder and also Prader-Willi syndrome and other child disability research. Thank you, Mary. So with all of the work that I do, I think of it in three themes. I want to use data to understand the effects of rare disorders, including Rett syndrome, and what can be modified. I want to know how to measure impact accurately and correctly, and I want to be able to provide effective interventions that improve domains that matter to families. Thank you, Mary. And so my next three slides are just three examples of work, and a big part of our work has been measurement. Measurement is the little sister to developing new treatments, and if we're going to have new treatments, we need to be able to measure them well, or we won't know really what they're doing. And so, as I said earlier, we have generated a gross motor measure for Rett syndrome and a hand function measure for Rett syndrome, and excitingly, these have been developed through working with families across Australia, but excitingly, they are both being used in the two gene therapy trials that are being tested right now. So that is very exciting translational sort of an opportunity for work from collecting data to go through this full pipeline of activity and validation and then be actually used in practice when we want it to matter. But the example on this slide, sorry, go back, Mary. The example on this slide relates to quality of life, and quality of life is really it encapsulates how we live, and we all want to live well with a strong quality of life. And this program of research has been driven by parents telling us that quality of life actually isn't a soft outcome for them or for their child. It's their most important outcome for their children and for their families. And accordingly, it should be everybody's business. And without a valid measure of quality of life for children, including for Rett syndrome, we developed the Quality of Life Inventory Disability. And this is a measure, it's a 32-item measure of quality of life. Children or parents of children with Rett syndrome were critical to its development, and it's now being used extensively across the world by families for goal setting, for NDIS planning, by clinicians in their goal setting. Some departments are using it for their benchmarking and planning. Registry studies such as the new International Rett Syndrome Foundation questionnaires are using QI disability, and it's being used in clinical trials internationally for children with rare genetic epilepsies. Thank you, Mary. The second example is a more than 12-year pipeline of research which looked at ways that wanting to know how we could help girls and adults with Rett syndrome to be more physically active. We started by learning how to measure steps in Rett syndrome, and some of you will have been putting on some of the devices that we will have posted to you for your children to walk around in. And from measurement, we then went to families to understand how we could encourage their daughters to be more active using focus groups and surveys. Looking at the bottom, we then developed interventions that we tested with a pilot study and a randomized controlled trial. And to the top right corner, you can see that very colourful banner. And we then worked with parents to develop a web page called Active Rett, which has lots of information on how to encourage your daughter to be more active. And you can see the web address at the bottom of the slide. Googling Active Rett will get you there. Next slide, Mary. And finally, this is a second pipeline of research, and this is well over 10 years. We started by doing consensus studies where we gathered international panels of clinicians and researchers who were expert in Rett syndrome and developed a set of guidelines for growth and nutrition and gastrointestinal disorders. Click again. We've collected other data that has taught us about the value of gastrostomy, and if it is recommended for a girl or an adult, it is generally acceptable. There are generally few complications, and we also know, as you can see in the graph, that hospitalizations will go down with time following gastrostomy, adjusting for age. And we also know from interview studies that there are advantages to quality of life. Could you click again? And then with all of that information, we were able to translate that into the development of a parent resource. And again, it was co-designed. It includes videos and worksheets and separated into modules on deciding about gastrostomy, the hospital stay, and then living with gastrostomy after you go home. We've called it Nourish, and it's published on the Developmental Disability WA website. You can see the web address for Nourish at the bottom of the page. And we were very proud, Telethon Kids, to partner with a community advocacy organisation to develop this resource, promote it and house it on their webpage. Thank you. I think that's my last slide.
Thank you very much, Jenny, for that. Yes, an amazing 50 or so minutes of quite a fair bit of information. Great brief, I think, to give everyone insight, and I thank you all for the insight you've given us this afternoon. I'll go on to have a quick chat about the Nashville conference. I had the pleasure to attend the Nashville conference along with Rett Granddad, who you met today, Eric Gowns, and in attendance there's also Associate Professor Wendy Gold, Associate Professor Carolyn Ellaway, Professor Helen Leonard, and Associate Professor Jenny Downs, all of whom you've heard from today. So it was quite an exciting meeting, and it will be one that will be, I suppose, etched in my memory forever out of all the meetings that I've been to, because on the first day, it was announced that the first Rett patient was dosed with gene therapy. So an amazing buzz you can imagine at a scientific conference at 9 a.m. when the doors open, and that's the first thing that sort of hit social media and the news. It was a very positive experience listening to all the talks and presentations. The knowledge in advancements, treating drugs and gene therapy was just incredible. And one of the things I noticed from this conference was the collaboration between drug companies, universities, labs was incredible. I hadn't seen any of this to any conferences I've been to before, and I've obviously only gone to Rett conferences, so I don't know outside of Rett syndrome if that sort of happens. But in the past, the amount of data that was shared that was unpublished, and there was challenges from quite high people to other labs to work together with them to advance the research and ultimately finding that gene therapy happy medium. The collaboration was fantastic. We did a fair bit of publicity for our Congress next year, and we made a lot of great connections, forged new alliances, and met various pharmaceuticals who we actually had a couple of meetings while there with the SMAC committee. So it was a wonderful, I suppose, contingent of Australians over there that represented Australia. And it really showed that Rett syndrome in Australia is at the forefront of research and technology. As I mentioned, there was 4 other people in this meeting there, so I'm happy if anyone else would like to mention some of the scientific stuff which sort of went over my head. But it was quite exciting times. No, all good. All right. On that note,
Sorry, Claude. I believe
Yes.
Eric, you were trying to speak, but you're on mute. You could just unmute.
Yes, I was just going to mention that we could mention that Carolyn, Wendy and I wrote a scientific report on the Congress that is written in lay terms that is now on the RSAA website.
Yes, thanks for that, Eric. And I think there was a link for that in the newsletter as well, which would take you to that, article. Thank you, Eric. All right, questions. I think I've seen one question from the group. Just bear with me one second. Okay, so I've got a question for Marianne. And the question is, one second. Oh. Your daughter has preserved speech. What does she talk about and what education has she had?
And I'll say this, knowing that most Rett parents would kill to hear their daughters speak. We often say that Jacinta basically never shuts up. So she speaks quite coherently. Her journey was interesting in that, as I said to you, she wasn't diagnosed until quite late. Her IQ regressed. She had some fairly typical hand movements with the sort of tapping, and she did a lot of self-mutilation. She started falling quite a lot. She's only ever had a couple of seizures. Her education ended up, because we didn't have the diagnosis, she actually went through a fairly normal education. She went to a normal primary school, a state primary, but it became apparent that she was falling behind in her education. Then we ended up sending her to a private primary, thinking we'd get a bit more support. Again, her IQ to continue to deteriorate, and we ended up sending her to a school called Rossbourne School in Melbourne, which you have to have an IQ of above 70. And at that stage, her IQ was just above 70. Her IQ now has fallen to 52. Then she ended up going and doing some education through TAFE. She never achieved a proper certificate. It all had to be very supported. But she still is relatively independent. She now gets herself to a supported work environment on public transport, and as long as everything's going well, it's fine. She can't plan. She has a very poor short-term memory, but she's got a wide variety of friends, and she has issues with eating. She dribbles quite a bit, for which she's been bullied over the years, especially before we knew what was going on with her. So her path has actually been relatively, I use the word normal, but you know what I mean. It's been a relatively good path for her, and she certainly talks about, she understands now that she has Rett syndrome, or atypical Rett syndrome, that she's a Rett girl, and she uses it. I remember the first time we sat down and spoke to her after her genetics were done, she actually said to us two things. She said, okay, so the way I am, it's not my fault. And we said, no, it's not your fault. And she said, and that means that I can't pass it on to anyone else, because she was worried that she had something that she was going to infect other people with. So she's got some understanding. She gets very frustrated, as you can imagine, because she's got a couple of siblings who are fairly high achieving, and we're not a very tolerant family most of the time. But so her development's been interesting, and her education so far, as I say, she's now in a supported work environment.
Thank you, Marianne. That's very interesting, because my daughter is much more disabled, but she's done her VCE, which is the equivalent of year 12, very slowly, a subject a year. And I used to take her around to the TAFE colleges. And she's done started a subject in university, not University of the Air, Open Universities Australia, and she did it at Murdoch, did her first subject, and she had a wonderful tutor, because we were getting very anxious by the end of the year that she wasn't getting the work done in time. And he said, don't worry, time doesn't matter, just do the best you can. And so she had, time extended. And she just loves education. It takes the place of not being able to make friends easily. And she used to keep asking me to find her friends, and I couldn't, because I'm old. I mean, she's going to be 59 in a month or two. So I think she's sort of given up that idea. We've got a close family circle, and that's the best we can do friend-wise. But of course, on the internet, no one knows you're a dog or whatever. they say. So she could presumably have pen friends, but
I think we've been very privileged to have children that function as highly, as much as we are frustrated for them. I think we're very privileged to have children that function as highly as they do.
It's very, very unusual. Thank you both for your insights on your children. I've got a question for Dr. Carolyn Ellaway, if I may. The question is, does anyone know if there's a particular mutation type which was more prevalent in the Anavex trials?
Thanks, Claude. So in regards to, firstly, the eligibility criteria, one of them was that you had to have a mutation in the MECP2 gene, but it didn't say any particular type of mutation, as long as there was a clinical diagnosis of Rett syndrome followed up with a confirmation of a mutation in the MECP2 gene. At the moment, all the data is about to be locked. So we don't have any information about the effects of the clinical trial, individual patient data. So I can't answer that question, but just suffice to say that you didn't have to have a specific mutation type to become part of that clinical trial or eligible for the clinical trial. And most clinical trials that I've been involved with, as long as you have a clinical diagnosis of Rett syndrome confirmed with a MECP2 mutation that is pathogenic, you potentially could be eligible. But obviously at the completion of the study, the data will then be unlocked and we'll have more information about the participants and the effect of Anavex in the cohort.
Thank you, Dr. Carolyn. While we're talking, Dr. Carolyn, I would like to ask if you could possibly give us a little bit more insight on the neurotech cannabis study that you are the principal investigator on.
Yeah, so this is just still early days. I'm looking to do a clinical trial of a full spectrum medicinal cannabis, so not just CBD, which was the other clinical trial, but most clinical trials in other groups are just CBD with the THC, which is a psychoactive component, removed. But this is a full spectrum medicinal cannabis. So in marijuana or cannabis, there are over 100 different cannabinoids. And so, as I said, this will be a full spectrum. There is currently a study going on in Melbourne in a cohort of children with level 2 to 3 autism, so quite severe autism. And in that study, there has been significant improvements in that cohort in their levels of anxiety and their communication. So the drug company reached out to me to see if I would be interested in a study in Rett syndrome, and of course I said yes. So we're at this stage just waiting on approval from the Human Research and Ethics Committee of the Sydney Children's Hospital Network. To start with, it will be a small cohort, mostly focused on New South Wales, but it's a short study, and then hopefully we'll be able to open it up, and it's likely there will be other sites in Australia. But I must say, I think this could be an interesting clinical trial to be involved in. I just want to let you know also that it's extremely early days, but I am also in communication with one of the drug companies who are interested in bringing a gene therapy trial to Australia as well.
Thank you, Dr. Carolyn. I've got another question here. I'm not sure who's probably the best person to answer it, but it may even be me, but any indications on when trofinetide would be available in Australia?
Do you want me to comment on that, Claude?
Yeah, if you like. I've got some insight, but I'll let you go ahead.
Okay, well, you feel free to go ahead if you've got inside information, because I don't have inside information.
Oh, okay. So from the information I have, at this stage, there's no definitive date on when it will be available. There's still quite a lengthy process that needs to be to go through before we get to that point. And I'm sure everyone can comment on that, or the SMAC committee can comment on that before that happens. So first and foremost, the biggest hurdle was finding a distributor and producer of the drug, which Neuren have since done, and that announcement took place last week with Acadia taking that on for the world. And the next process is for Acadia to make applications in relevant countries to have the drug approved and going through the local approval processes. From what I have been told, that is at least 18 months away before any of that process happens.
Do you mind if I now comment, Claude?
Yes.
So I just want families to appreciate you're not missing out. Even though there has been FDA approval in America for trofinetide, one of the presentations at the Nashville conference was in relation to the clinical trials. And one of the concerning things, particularly from my perspective as a clinician caring for children with Rett syndrome, is the high frequency of quite a significant side effect being diarrhea. And as you know, girls with Rett syndrome generally have significant constipation. So this is quite extreme for girls with Rett syndrome. Diarrhea was reported in approximately 70 to 75% of the cohort, and about 25% of the cohort lost weight. So I have a few little concerns. What I still don't quite understand from what was presented is, if I prescribe this medication, what is the effect on an individual with Rett syndrome? There was talk about different questionnaires and significant improvements, but I still don't have a very clear understanding of what I'm going to see if I prescribe the medication. So I still think something like that, it's early days. I'd really like to see a bit more data in relation to trofinetide, but I would like families not to feel like you're missing out because it hasn't been approved in Australia.
Thank you very much for that insight, Dr. Carolyn. I've got another question here in relation to genetics, so possibly either John or Helen. So John, is it possible to have any idea how many girls are out there with MECP2 mutation who are carriers? Maybe it's not as rare as we think, given the non-affected gene could be one being expressed.
Yeah, thanks for that. So when the term carrier is used, I'm presuming that means an individual carries an otherwise a change in the MECP2 gene that would otherwise cause disease, but that individual herself does not have any manifestations of the disorder. So that's a pretty rare phenomenon based on our understanding. In fact, in most of the time, individuals with who have Rett syndrome caused by MECP2 mutations, the mutation has arisen as a mistake at the time of conception, and so hasn't been passed down from a parent. There are some very rare exceptions to that, but they are in the vast minority. Where there are individuals who have passed the faulty gene on to their daughters, so women who passed it on to their daughters, some of them have actually had mild or milder features that could be compatible with a less severe progression of Rett syndrome, and very few have been otherwise completely normal. And one of the factors that we think contributes to them escaping major harm is so-called skewing of X inactivation, which is a peculiar process to the X chromosome. So girls and women have two copies of the X chromosome, and usually one of them is switched on and one of them is switched off. And it's possible that there are, well, we know that there are individuals where that's highly skewed. So most of the time, one of the X chromosomes is switched on and the other one is switched off. And if it happens to be that the copy that is carrying the MECP2 mutation is the one that's switched off most of the time, then that will mean that these individuals have either mild or no clinical features. So I guess the short answer is yes, there could be some carriers out there, but based on our experience, we think that number is very low, and probably not very many are being kind of missed, if you like.
Thank you very much for that, John. Okay, on that note, if there are no more questions, there doesn't seem to be, I'd like to thank everybody for their participation, particularly our SMAC committee. Wonderful to have you on board, and it is a very exciting time for the Rett world. There's quite a fair bit happening on the horizon. At this point in time, there are over 20 companies that are invested in some form of study or clinical trial across the world. Obviously, we've just heard that there has been a patient dosed with gene therapy. We have another approved drug most recently this year, and there is a lot more drugs in the pipeline, as well as announcements of other gene therapies that will take place around the world. So quite an exciting time for Rett syndrome across the world. Going into next year, Australia, it's finally back on after almost 8 years. The 9th World Rett Syndrome Congress will be hosted by the Rett Syndrome Association of Australia on the Gold Coast. Will take place between the 2nd and 5th of October. That's Wednesday through to Saturday. We're still working on the venue, but it'll be very central to Surfers Paradise. The event will be a world-class event. So this is the opportunity for families to come out and meet all these professors from around the world, all these researchers. We'll be rubbing shoulders with people that have made discoveries. There'll be participation from a lot of the companies that have contributed to the research, which will give us the opportunity to speak firsthand. So the Congress will be running basically 2 streams. Being a World Congress, there is a commitment for scientists to get together and share data. So there'll be a purely scientific stream for data. And there'll also be a clinical stream for mums and dads to know about clinical innovations, therapies, and things like that. So again, looking very forward to this. There's still a little bit to go. The Rett Syndrome Committee is working hard to present this. We've got the SMAC committee on board. This is going to be a massive event in October next year, and we're hoping that you can plan a holiday with your family. So it's right smack in the middle of school holidays. Spend some time on the Gold Coast and attend this event. Updates will be posted on social media and in our newsletters, and the website is live. We expect to open the registrations later this year, and we'll start adding information to it as we have it at hand. So the program is well underway, as I mentioned earlier. There's still a bit of work there to do, but there'll be some quite renowned speakers that will be presenting to us here in Australia. All right. On that note, once again, I'd like to thank everybody that's attended here today, and a big thank you to Mary, who's pulled all this together. She's done an absolute amazing job. And I know we've gone over time, so I apologise for that, but it's a lot of information to try and squeeze into an hour. All right. Good afternoon all, and thank you so much. We'll see you again at the next webinar. Thank you. Bye.
Thanks, Mary and Claude.
Thank you.